8.3 Cell Structure and Functions. Biology is the study of living organisms. The detailed description of their forms and appearance only brought out their diversity. It is the cell theory that emphasized the unity underlying this diversity of forms, that is, the cellular organization of all life forms. A description of cell structure and cell growth by division is given in the chapters comprising this unit. Cell theory also created a sense of mystery around the living phenomena, that is, physiological and behavioral processes. This mystery was the requirement of integrity of cellular organization for living phenomena to be demonstrated or observed. In the studying and understanding the physiological and behavioral processes, one can take physiochemical approach and use the cell-free systems to investigate this approach enables us to describe the various processes in molecular terms. The approach is established by the analysis of the living tissues for elements and compounds. It will tell us what types of organic compounds are present in living organisms. In next stage, one can ask the question, what are these compounds doing in the cell? And in what way they carry out the gross physiological processes like digestion, excretion, memory, defense, recognition, etc. In other words, we answer the question, what is molecular basis of physiological processes? It can also explain the abnormal processes that occur during any diseased condition. This physiochemical approach to study the and understand the living organism is called the reductionist biology. The concept and techniques of physics and chemistry are applied to the concepts, and techniques of physics and chemistry are applied to understand biology. In the chapter 9 of this unit, a brief description of the biomolecule is provided. Chapter 8 Cell, the unit of life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is that makes an organism living? Or what is that an inanimate things does not have which a living thing has? An answer to this is the presence of basic unit of life, the cell, in the all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called the unicellular organisms, while others like us composed of many cells are called as multicellular organisms. 8.1 What is cell? Unicellular organisms are capable of, first, independent existence, and second, performing the essential function of life. Anything less than a complete structure of a cell does not ensure the independent living. Hence, cell is the fundamental, structural and functional unit of the all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and the improvement leading to the electron microscope revealed all the structural details of the cell. 8.2 Cell Theory. In 1838, Matthias Sheldon, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissue of the plant. At about the same time, Theodore Shevin, the British zoologist, studied different types of animal cells and reported that the cell had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on the plant tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Shevin proposed the hypothesis that the bodies of animals and plants are composed of cells and product of cells. Sheldon and Shevin together formulated the cell theory. The theory, however, did not explain as to how new cells were formed. Rudolf Virchow first explained that the cells divided and new cells are formed from the pre-existing cells, that is omnicellula, e-cellula. He modified the hypothesis of the Sheldon and Shevin to give the cell theory a final shape. Cell theory as understood today is, first, all living organisms are composed of the cells and product of cells. Second, all organisms arise from the pre-existing cells. 8.3 An overview of cells. You have earlier observed cells in an onion peel and on a human cheek. Cells under the microscope. Let us recollect their structure. The onion cell, which is the typical plant cell, has the distinct cell wall and its outer boundary, and just within it is the cell membrane. The cells of a human cheek have an outer membrane as a delimiting structure of the cell. Inside each cell is the dense membrane bound structure called the nucleus. This nucleus contains a chromosome which in turn contact the genetic material, DNA. Cells that have membrane bound nuclei are called the eukaryotic, whereas the cells that lack the cell membrane bound nucleus are called the prokaryotic. In both prokaryotic and eukaryotic, semi-fluid matrix called the cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of the cellular activities in both plant and animal cells. Various chemical reactions occur in it to keep the cell in a living state. Besides the nucleus, the eukaryotic cells have the membrane-bound distinct structure called the organelles like endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelle found in the cell of the both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles of the chloroplast, that is in plants, and mitochondria, and on the rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called the centrosome, which helps in the cell division. Cells differ greatly in the size, shape, and activities. Figure 8.1. For example, mycoplasmas, the smallest cell, are only 0.3 micrometer in the lengthwise, while the bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of the ostrich. Among the multicellular organisms, human red blood cells are about 7.0 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in the shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even the irregular. The shape of the cell may vary with the function they perform. 8.4. Prokaryotic cells. The prokaryotic cells are represented by a bacteria, blue-green algae, mycoplasma, and PPLO, that is pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, means rod-like, coccus, means spherical, vibrio, means comma-shaped, and spirillum, means spiral. The organization of the prokaryotic cell is fundamentally similar, even though the prokaryotes inhibit a wide variety of shapes and functions. All the prokaryotes have the cell wall surrounding the cell membrane except in the mycoplasma, the fluid matrix filling the cell in the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by the nuclear membrane. In addition to genomic DNA, that is the single chromosome or a circular DNA, many bacteria have the small circular DNA outside the genomic DNA. These smaller DNA are called the plasmids. The plasmid DNA confers the certain unique phenotype of the characters of the such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor the bacterial transformation with the foreign DNA. Nuclear membrane is found in the eukaryotes. No organelles like the ones in eukaryotes are found in the prokaryotic cells except for the ribosomes. Prokaryotes have the something unique in the form of inclusions, a specialized differentiated form of the cell membrane called mesosome. Mesosome is characteristic of the prokaryotes that they are essentially infoldings of the cell membrane. 8.4.1 Cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have the chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs a distinct function, they act together as a single protective unit. Bacteria can classified into two groups on the basis of the differences in the cell envelope and the manner in which they respond to the staining procedure developed by the gram, which is those that they take up the gram stain are gram positive and others do not, are called the gram negative bacteria. Glycocalyx differs in the composition and thickness among the different bacteria. It could be loose sheath called the slime layer in some, while in the others may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides the strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is a selectively permeable in nature and interacts with the outside the world. This membrane is similar structurally to that the eukaryotes. The spatial membranous structure is the mesosome, which is by the extensions of the plasma membrane into the cell. These extensions are in the form of vesicles, tubules and the lamellae. They help in the cell wall formation, DNA replication and distribution to the daughter cells. They also help in a respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contains pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show the range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook and the basal body. The filament is the longest portion and extends from the cell surface to outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play role in the motility. The pili are a long tubular structure made up of the spatial protein. The fimbriae are small bristle-like fibers sprouting out the cell. In some bacteria, they are known to help attach the bacteria to rocks in the streams and also to the host tissues. 8.4.2 Ribosomes and the inclusion bodies. In prokaryotes, the ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer to 20 nanometer in size and are made of two subunits, 50S and 30S units, which when present together form a 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to the single mRNA and form a chain called the polyribosomes or a polysome. The ribosome of a polysome translate the mRNA into proteins. Inclusion bodies. Reserve material in the prokaryotic cell are stored in the cytoplasm in the form of inclusion bodies. These are not bounded by any membrane system and lie free in a cytoplasm. Example: phosphate granules, cyanophycine granules and glycogen granules. Gas vacuoles are found in the blue, green and purple and green photosynthetic bacteria. 8.5 Eukaryotic cells. The eukaryotes include all the protist, plants, animals and fungi. In eukaryotic cells, there is an extensive compartmentalization of the cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess the organized nucleus with the nuclear envelope. In addition, the eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into the chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as a former possesses cell walls, plastids and large central vacuole which are absent in the animal cells. On the other hand, animal cells have the centrioles which are absent in the almost all plant cells. Let us now look at the individual cell organelle to understand their structure and functions. 8.5.1 Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially on the human red blood cells (RBCs), enabled the scientists to deduce the possible structure of the plasma membrane. The studies showed that the cell membrane is mainly composed of the lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of a saturated hydrocarbons is protected from the aqueous environment. Figure 8.4. In addition to phospholipids membrane also contain the cholesterol. Later, the biochemical investigation clearly revealed that the cell membranes also possesses the protein and the carbohydrate. The ratio of protein and lipid varies considerably in the different cell types. In the human beings, the membrane of the erythrocyte has approximately 52% of the protein and 40% of lipid. Depending on the ease of extraction, the membrane proteins can be classified in the integral and the peripheral. The peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of the cell membrane was proposed by Single and Nicholson in 1972, widely accepted as a fluid mosaic model. Figure 8.4. According to this, the quasi-fluid nature of lipid enables lateral movement of the proteins within the overall bilayer. This ability to move within the membrane is measured as a fluidity. The fluid nature of the membrane is also important form of a point of view of functions like cell growth, formations of the intercellular junctions, secretions, endocytosis, and the cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on the either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called a passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from the higher concentration to lower. Water may also move across the membrane from the higher to lower concentration. Movement of the water by the diffusion is called osmosis. As the polar molecule cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is, from the lower to higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called the active transport. Example, Na plus K plus pump. 8.5.2 Cell wall. As you may recall, a non-living rigid structure called a cell wall forms an outer covering of the plasma membrane of the fungi and the plants. Cell wall not only gives shape to the cell and protects the cell from the mechanical damage and infection, it also helps in the cell to cell interaction and provides the barrier to undesirable macromolecules. Algae have the cell wall made up of the cellulose, galactans, mannans, and minerals like calcium carbonate, while other plants consist the cellulose, hemicellulose, pectins, and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner or towards membrane side of the cell. The middle lamella is the layer mainly of calcium pectate, which holds or glues the different neighboring cells together. The cell wall and the middle lamellae may be traversed by the plasmodesmata, which connect the cytoplasm of the neighboring cells.